All right, this mini lecture is going to be about the cell. And amazingly, we're going to talk about aquaporin synthesis. So we're going to keep talking about this protein as an example of a protein, one of several tools that are used in a cell. And aquaporin is a cell membrane protein. So we're going to have to talk about how aquaporin gets made and then gets transported to the cell surface. It's a cell membrane protein. It's called a transmembrane protein. That means it crosses the membrane. So here is the phospholipid bilayer that is the cell membrane, like that, and aquaporin crosses the membrane. It's on both sides of the membrane. It has a cytoplasmic side, the intracellular side, and it has an extracellular side. And inside the cell is the cytoplasm. And so it's a transmembrane protein, or sometimes it would be another synonym for that is it's a membrane spanning protein. So we're going to talk about how it gets made, and we're talking about it in this context because, as you can see here, that in Chapter 6 there are lots of names for us to learn. And there are tables of names and lists of names, and we should try to tell stories in order to help provide some framework. So that's what we're going to do with this mini lecture. We're going to tell a story. So it's going to be a story that takes place in plant and animal cells that make aquaporin. And in general, in our course, we emphasize generalities in this intro course because we're learning basic principles. And so this story is not going to be one that takes place in all cells. This is going to be taking place in eukaryotic cells, and eukaryotic cells, or eukaryotes, are plant and animal cells, and also single-celled eukaryotes like yeast and other fungi like that. And eukaryotes are not bacteria. Bacteria and blue-green algae fit into a large class called prokaryotes. So as you can see here, there are lots of things that have in common between eukaryotes and prokaryotes, these and many more, but there's some differences. And eukaryotes means it has a true nucleus. It has a nucleus inside the cell that is surrounded by a membrane, and the nucleus contains the DNA. Prokaryotes have DNA, but it's not surrounded by a nuclear membrane. So when we talk about this, we're talking about lots and lots of life, but not all life, because we're going to be really, this chapter really focuses on eukaryotes. And then we want to also keep track of our scale. So remember that this is a cell that we're talking about, and we're talking about stuff happening inside the cell. And there'll be lots of talk about the trucks, the different kinds of trucks, proteins within the cell, because the proteins within the cell really provide function. And we're going to talk about aquaporin. And there are tens of thousands of aquaporin proteins on the surface of each cell in our body, because all cells need to let water in and out for a variety of reasons, and they need to let it in and out really fast, faster than it would trickle across the cell membrane phospholipid. So we got a special channel. Here's a figure from your book, a table from your book, and you can see that with this table there is just a lot of potential memorizing. And so with this video we're going to try to start to break through some of that and tell some stories so that maybe you won't have to do so much kind of just, well, I got to memorize this list. Okay, and it goes like this. Oh, we're going to look at the synthesis of aquaporin, and then after we do that, we're going to make a quick comparison with insulin and hemoglobin, and then another protein. So we've got 4 different proteins that we're going to talk about getting synthesized in a eukaryotic cell. Imagine a human cell. And so there's just some principles. All cells have lots of proteins, and each performs a specific function based on its structure. All proteins are made by ribosomes. all proteins in prokaryotes and eukaryotes. And so ribosomes are inside of every cell, and ribosomes are going to be synthesizing proteins. So the site of protein synthesis in all cells are these ribosomes. And what's the function of the ribosome? The function of the ribosome is chaining together amino acids by a covalent peptide bond. And how do they know? How do the ribosomes know which amino acid to add? Because they're going to be using an mRNA template. And so the mRNA template has the code. The mRNA template is copied from the DNA. And the DNA will have a gene for aquaporin with this coding information available on it. We get copied into RNA, and then ribosomes use that RNA template to tell which of the 20 amino acids to add in sequence to make a human aquaporin. So, as we've said in videos a couple of times, eat proteins, they're broken down into amino acids and then delivered to each cell. Each cell has to use its ribosomes to synthesize the proteins it needs. There are two basic types of proteins we could talk about that get made in our cells by ribosomes. There are proteins that are secreted from the cell or bound to the cell membrane, and one of the examples of that is aquaporin. And then there are proteins that float around in the cytoplasm. So let's go back to this image and say that there are some cells that are, there are some cells that have ribosomes that make proteins and basically do two different types of protein synthesis. And one protein synthesis is where the ribosomes just synthesize proteins that float inside the cell. Cytoplasm. The other type of protein is a protein like aquaporin, proteins bound to the membrane, bound to, I could say, membranes. It's like the cell membrane, but also there are membranes throughout the cell that we'll learn about. And so proteins bound to membranes are released from the cell. Some proteins get secreted or released from the cell. So they head out. So we have these two basic types of proteins, and they're both synthesized by ribosomes. Ooh, I don't know why I have that there. Oh, I backed up, that's why. So here we go with this. So proteins that are bound to the cell membrane or secreted include aquaporin, insulin, proton pumps, and the LDL receptor. And I underlined those three because I said we're going to learn about those three, and then proteins that float around, we'll learn about that one in our next little list. Okay, so we're going to start out following the synthesis of aquaporin as our first example of those. And it's a figure from your book, and in the nucleus of a eukaryotic cell, there is DNA, and the DNA is inherited from our parents and is the source information to code for proteins. That's a big job of DNA, is to hold information that can be used to code for proteins. That means tell the ribosome which amino acid to add in this long chain. And to do that job, the DNA is first copied into mRNA. The mRNA leaves the nucleus and is bound by the ribosome, and is used by the ribosome as a template to tell which amino acid to add next. Okay, so in the cytoplasm, we have a ribosome. There are thousands of ribosomes in the cytoplasm, and the first step of ribosome protein synthesis is the ribosome binds an mRNA, and then it starts to copy that mRNA into protein, using the RNA as a template, an indication of which amino acids to add. The amino acids start to get joined together, and there are some proteins that at the very beginning of the protein, there is about a 20 amino acid sequence that is called a signal peptide. And aquaporin has a signal peptide, and The signal peptide is what targets secreted proteins and membrane proteins to get to their proper location. So what happens is the ribosome is just loose in the cytoplasm. It starts to synthesize the protein, and if the first amino acids are a signal peptide or signal sequence, the ribosome stops synthesis and it just floats until it bumps into special organelle in the cell called the rough ER. Rough ER stands for endoplasmic reticulum, and there are two types in the cell. There's the rough ER and the smooth ER. We're talking about the rough ER right now, and the rough ER is an organelle. An organelle is a membrane-bound region within a eukaryotic cell. So eukaryotes have organelles, and prokaryotes generally do not have organelles. There are a couple exceptions to that, but generally prokaryotes not in prokaryotes. Membrane-bound region of a eukaryotic cell. So we already learned about the nucleus, and the function of the nucleus is its storage site for DNA and the site of mRNA synthesis. Then the ribosome is considered an organelle, even though it's not bound by a membrane, no membrane. And the ribosome's function is the site of protein synthesis. Then the next is the rough ER, and the rough ER is the site of protein synthesis for proteins that are going to be bound to the membrane or secreted from the cell. So here's the rough ER, and the rough ER is called rough because there are ribosomes on its surface. Ribosomes on its surface because ribosomes that start to translate a mRNA that has a signal sequence stop synthesis until they bind and dock at the rough ER. And then they continue synthesis. And so let's just go to a place where we have some space and say for aquaporin, aquaporin starts to get synthesized by the ribosome, and aquaporin has a signal sequence that leads the rough ER, leads this ribosome to get bound to the rough ER, and then continue and complete synthesis of aquaporin, which is a donut that will just float in the cell membrane. And where aquaporin is first made is not at the cell membrane where it has to do its job. Where it's first made is on the rough ER. So now we know the function of the rough ER. That's to make, it's the site of protein synthesis for proteins that are bound to the membrane or that are going to get released from the cell. And so proteins that would be floating in the inside of the rough ER, which is called the lumen, that's the space inside the rough ER. Floating here would be proteins like insulin. Insulin is going to get released from the cell into the blood. And the site of protein synthesis for aquaporin and insulin and lots of other membrane-bound and secreted proteins is the rough ER. So the ribosome binds and the signal sequence is removed. It's not part of the mature protein, and then synthesis is completed. And now we have aquaporin synthesized, and remember that it's human aquaporin because the food we eat contains proteins that are aquaporin, but those get digested to individual amino acids, and then the amino acids carried to each cell, and each cell has this process going on. Protein synthesis is like an assembly line. Proteins get used up over time and have to be replaced. So at a low level, if a cell has a protein, it must be continuing to make it. All right, so the next thing is to say that here's the pathway for this newly aquaporin to get to the cell surface. Okay, so initially the protein is begun to be synthesized by ribosome, just floating in the cytoplasm. Then the signal sequence, signal peptide, causes the ribosome to be delivered to the rough ER, and then you have the rough ER has a membrane, so this is a rough ER, and there's a membrane, and the membrane's a phospholipid, and it now has donuts of aquaporin floating in it. And aquaporin, remember, is floating in the membrane because it has a belt around its outside that is hydrophobic amino acids. So the aquaporin is bobbing in the membrane, stuck in the membrane, because outside the cell is a water environment and inside the cell is a water environment. And this stripe around the waist of aquaporin on the outside, the belt, is hydrophobic. And so I drew over here, that's the cell membrane phospholipids. So we've got a protein that is floating and bobbing in the cell membrane, but it's also not going to go anywhere. And so to get this to the cell surface, what happens is that the rough ER makes a little bubble like that. Some aquaporin are in the membrane of that bubble, and the bubble pinches off and captures some aquaporin like that. The aquaporin then is delivered, and this is called a vesicle. The aquaporin gets delivered to its next stop, and that's an organelle called the Golgi apparatus. Golgi apparatus. And the little vesicle fuses with the Golgi, like two bubbles coming together, so that initially when it fuses it looks like this, and then later like this, and then later it's just part of the cell membrane. So the newly synthesized aquaporin ends up getting delivered to the membrane of the Golgi. And in the Golgi, some changes get made. Some proteins are glycoproteins, and a little sugar polysaccharide is added to the protein to become part of the protein. Other proteins get cut and modified in the Golgi, and eventually they pass out of the Golgi by pinching off another vesicle, and that vesicle is taken by some, on like a train track system. The little vesicle is carried to the cell membrane, and then the membrane fuses with the cell membrane to give us what is this vesicle, and phospholipids and proteins from the Golgi eventually fuses with the cell membrane, and now part of the cell membrane is this vesicle, the phospholipids of this vesicle. And the part of the membrane are the proteins that are brought. Notice that when the vesicle fuses in a process that's called exocytosis, that when it fuses, it's now like this process of two membrane-bound regions fusing together. And so we can just draw this a little bit more. The vesicle arrives at the cell membrane, and exocytosis involves the docking of the vesicle right on the cell membrane, and then to bring together and fusing the membrane so that we would go from phospholipid bilayer that looks like this to phospholipid bilayer that looks like that. So both phospholipid bilayers, but just some different molecules composing them. And once the exocytosis has occurred, the membranes really fuse together like that and become part of the same thing. So we're delivering the protein, we're also delivering phospholipids. So this pathway includes some important concepts. One important concept is exocytosis. And exocytosis is a way to deliver proteins or phospholipids that have been gotten made, and the membrane is changing its composition. The delivery proteins could include the membrane-bound proteins, like we just learned about, or very easily a secreted protein. So let me just draw this again and say that the vesicle has aquaporin in its cell membrane and lots of other proteins there. And then it could have insulin in the interior of the vesicle, the lumen of the vesicle. When that fuses with the cell membrane, right, we're going to now have new phospholipids in the cell membrane. We're also going to have aquaporin protein delivered to the membrane, eventually diffusing throughout the membrane like that. This is the cell membrane. And notice that if there's a protein like insulin that's floating in the lumen, that protein is now going to be released into the blood or this extracellular environment. And so just like that, we've described the synthesis of aquaporin and insulin. Insulin has a signaling peptide, a signal sequence that helps the ribosome start and then dock at the rough ER to complete the synthesis of insulin at the rough ER. And then eventually processing it through the Golgi. A lot of different things can happen there, and then it gets released from the cell. That's good to compare to a protein like hemoglobin. We just draw immature red blood cell. Here's a hemoglobin. Here's a ribosome. And there's a nucleus. And in the nucleus is DNA. And the DNA includes DNA for all the different genes, including these genes, like that. And so the gene is going to get copied into RNA. The RNA is going to come out, diffuse out of the nucleus, and be bound by a ribosome. The ribosome will copy it. And in the case of aquaporin and insulin, there's a pause to get the ribosome delivered to the rough ER so that the protein gets made in the proper location. In the case of hemoglobin, there's no pause because when hemoglobin gets made, the hemoglobin protein just floats in the cytoplasm of the immature red blood cell. And so that's all there is to it. The third example is easy as pie. And that is that hemoglobin is synthesized by ribosomes that don't find a signal sequence and just complete synthesis and release it into the cytoplasm. And for all these proteins, there are thousands and thousands of copies of them in each cell. Then the last comparison I'll make is to a lysosomal proton pump. And what's going there is that big cell, and there's an organelle called the lysosome. And the lysosome is the cell's stomach. It functions like the cell's stomach to digest, to break down, metabolize materials. And it will be a variety of things. But one thing that happens is that if a protein gets made inappropriately, that protein can get taken to the lysosome and degraded. The lysosome has a very low pH, very acidic pH, that means very high concentrations of the hydrogen ion. And the way that occurs is there are transmembrane proteins in the membrane of the cytoplasm of the lysosome, and those proteins use energy to pump hydrogen ions into the cell. Into the organelle, the lysosome. So, where do those proton pumps come from? The answer is they're synthesized. Where are they synthesized? The answer is ribosomes. And they are transmembrane proteins. So they're floating in a membrane, and they would want to be in a membrane right from the start when they're synthesized. So the lysosomal proton pump gene codes for an RNA. So the DNA gets copied into RNA, and that RNA is bound by a ribosome, and the ribosome runs into a signal sequence. And in the case of lysosomal proton pump, the mRNA is delivered to the rough ER, and then synthesis continues. And a proton pump is in the cell membrane, and it will get pinched off through a vesicle, and that has the proton pump in the cell membrane. Then that will fuse with the Golgi apparatus. Some processing will get done, and then a vesicle will head to the cell surface. No, big point here. Instead of heading to the cell surface, the lysosomal protons, pump proteins, are targeted to the lysosome. So after the Golgi, there is an option. And lysosomal proton pump proteins get modified in the Golgi by having a sugar put on that includes a 6-mannose phosphate molecule. 6-mannose phosphate, and proteins that have that 6-mannose phosphate, that is used as a tag to tell them to be delivered to the lysosome, like that. So here we have this idea of vesicle trafficking, bringing stuff to the cell membrane, and we also have the idea of vesicles turning and heading to different directions depending on the cargo they're carrying. Here's just a drawing of this secretory pathway. In this case, this little green would be the equivalent of a protein like insulin that's going to eventually be released, free to float away from the cell, like that. I won't read through this. This is just some summary of aquaporin synthesis. And like I said, have emphasized that this isn't all cells. Bacteria don't have aquaporin, but it is lots and lots of cells. All plants and animals have it, and this is how that protein gets made. So we talked through that protein as an example of transmembrane protein, then talk through insulin, about a secreted protein, then talk about hemoglobin as one example of thousands that are just free diffusing in the cytoplasm. Then talk about lysosomal proton pump to talk about a protein that's going to a different spot. And in doing so, let me just scroll back here. Woo, there it is. So, in doing so, we've learned in this story about lysosomes, about the Golgi and the function of the Golgi. We learned about the ER. We learned about ribosomes. We learned about the nucleus. Boom. And so there's a lot of information here, but if you could tell one good example of the function of each of these, we would be doing great. And so I'm going to stop this recording at this point.